are going to understand and proceed with the next part, which is the pathogenesis of viral infections, of rabies virus. The learning objective that by the end of this lecture, you should determine the mode of transmission and mechanism of infection of rabies, to propose a diagram to clarify the pathogenesis of rabies disease. What are the modes of transmission of rabies and the mechanism of infection? First of all, there is the step of viral inoculation, where the virus is introduced into the body, the human or animal body. The virus is inoculated into the body through the bite of a rabid animal, which sheds the virus in the saliva. This is the most common route of transmission, is through the bite of a rabid animal. It is a zoonotic disease. Infection occurs from animals to humans. The bite of a rabid animal, shedding the virus, transmit the virus from the animal to humans. Also, infection may occur through transdermal inoculation or through wounds. For example, if you have a cut in your skin or a wound of the skin and you get exposed to the virus-infected saliva, the virus enters and starts the process of infection and pathogenesis. Direct contact with the infectious material through abraded skin or intact mucous membrane. It is very important to notice here that the virus cannot penetrate intact skin unless there is an opening, a wound, through either the wound, a cut on the skin, or through the bite of the infected animal. So intact skin cannot be penetrated or invaded by rabies virus. There should be a cut in the skin in order that the virus could be introduced and starts the process of infection. Or intact mucous membrane. So the virus can enter through abraded skin or intact mucous membranes. How does it happens to occur or reach the mucous membranes, the intact mucous membranes? Either by contact or splashing of infected saliva, or by through the mouth, whatever the source of contact is. Very rarely inhalation and human-to-human transmission occurs. But the most common is through the bite. Number one, the bite of a rabid animal, shedding the virus in the saliva. Secondly, through transdermal inoculation through wounds. Direct contact with the infectious material through abraded skin and or intact mucous membrane. That's why I've underlined the word abraded skin and intact mucous membrane, to be clear enough. This is followed by, once the virus is introduced into the body, this is followed by a process of initial virus multiplication in the muscles, locally, or in the connective tissue. So once the virus is introduced through the wound or through the bite or whatever the method of transmission, it starts to replicate in the muscles and the connective tissue surrounding. At this stage, of course, no symptoms are obvious on the patient. The patient does not manifest any manifestation of the disease. At this stage, the virus is still replicating to increase in number and allow to be propagated in the body. The virus replicates in the muscle. This is the initial viral replication. And then at this stage, it is susceptible, it does not stimulate the immune response yet, because it is just preparing itself for the process of infection, but not ready for full infection and full disease manifestation. So it replicates, it does not stimulate the immune response so far, but susceptible to neutralization by rabies immunoglobulin. And it is capable of virus replication, can be depressed and preventing it from reaching the CNS. We stop here for a while. This means that when the virus is replicated locally in the muscle and the connective tissue, no symptoms appear on the patient. However, it does not stimulate immune response, but if you inject immunoglobulins, rabies immunoglobulins, to the site of infection or the site of the bite, these rabies immunoglobulins or specific immunoglobulins for rabies is capable or is able to neutralize the virus from reaching the CNS and starting the process of infection. And this is the key point we're going to see later for the prophylaxis and treatment of case of bites of infected animal. There is one important issue, that the virus does not reach the bloodstream. The virus could never be isolated or reaches the bloodstream. So if you take the blood sample and try to look for the virus, it could not be detected because it never reaches the bloodstream. Once replication has occurred, the initial replication occurs, the virus starts to attach to the susceptible receptors. Any viral replication, the virus, to produce infection and pathogenesis, it should connect or adsorb to susceptible receptors present on the susceptible host cell. The virus attaches to special nicotinic acetylcholine receptors present at the neuromuscular junctions. Then the virus travels through axons of the peripheral nerves through retrograde axonal transport. What is meant by retrograde axonal transport means that it travels to the nerve through the axons to reach the spinal cord. In the spinal cord, it replicates in the motor neurons of the spinal cord and the dorsal root ganglia. The virus is highly neurotropic, means it travels through the nerves but never reaches the bloodstream. So the virus is highly neurotropic or exclusively neurotropic, and it binds to special nicotinic acetylcholine receptors at the neuromuscular junction, travels through the axons of the peripheral nerves by retrograde axonal transport to reach the spinal cord, where it replicates in the spinal cord. So the primary initial replication occurs at the muscle, at the muscular and connective tissue. The second replication stage occurs in the spinal cord and the dorsal root ganglia, and then the virus reaches the brain, ascends through retrograde axonal transport to reach the brain. In the brain, it replicates in the neurons of the brain, causing to their destruction and fatal encephalitis. After this fatal encephalitis or destruction of the brain cells, the virus starts to spread through the peripheral nerves to the several organs in the body, mainly the salivary glands, the submaxillary salivary glands, where it replicates once more in the salivary gland, submaxillary salivary gland, and the virus is excreted intermittently in the saliva of the infected individual or animal. Other organs can be where rabies has been found include the sensory nerves around the skin of the hair follicles, the cornea, and in the pancreas. Here we can see the virus, after replicating in the brain, it descends through the sensory nerves to reach the skin surrounding, innervating around the hair follicles. So the virus is present in large amount around the hair follicle through their descent or reaching it through the sensory nerves, innervating the skin. So if we like to summarize what we have said, the process of pathogenesis, that the virus is transmitted mainly through the dog bite or through cut in the skin, through abraded skin, through intact mucous membrane. Here you can see a dog bite, and then it spreads to the muscle, where it replicates in the muscle and attaches to specific acetylcholine receptors at the neuromuscular junction. When it has replicated, it goes up through the peripheral nerves to reach the spinal corn. There occurs once more replication, and the virus ascends to the brain. In the brain, it replicates, produces destruction of the brain neurons, and then goes down, descends through the sensory nerves and peripheral nerves to the skin surrounding the hair follicles. Also can be present in the salivary glands, spreads to the salivary glands, spreads to the cornea, where it can be detected in large amount. Thank you.